want to say a little bit about the MHC that I didn't say last time. One of the themes of the class that I've tried to stress a lot is the connection between technology and the way things are and our abilities with technology. And this refers to MHC, and of course, the practical stuff for MHC is transplants, matching MHC for individuals, for humans, basic work in mice. And I also wanted to recognize a couple things here. People had technology, had antibodies against MHC products long before sequencing DNA was easy. Okay, so the original haplotypes, and remember we speak of haplotypes because the whole set of genes tend to be inherited together. Little recombination between them in man or mouse. Okay, man or woman or mouse. Thus, if you know the allele at a particular locus in the MHC of a human or a mouse, then you either know for sure or in good likelihood what the allele will be at a different locus. And also, so combining those two things, the MHC human genes were originally serologically defined by antibodies, such as you may have heard of DR4 or DR3. So referring to, and those were defined by antibodies that recognize a DR gene product, okay? Subsequently, as time elapsed, sometimes people were able to find fine specificities. They could divide DR4 into other groups. Like there was something called DR4W4, little 4, workshop 4 for groups defined when they paid closer attention. But just note that originally those haplotypes were serologically defined because that was the technology. So people said, you know, so-and-so is DR4. Now, then with the, and remember, this reflected though that there are multiple genes. For instance, in the human, I'm more familiar with human in class 2, there is DP, DQ, and DR, and if I recall correctly, there's roughly 10 times the expression of DR versus DQ, and then more DQ than DP. But that means is that all these genes are present, all these gene products are present, but might be defined by an antibody against DR4. Subsequently, up into the modern time now, we have the ability to sequence a lot and quickly. So we can look at what genes are present at not only the DR locus, but the DP and DQ. And not to give you too much minutia, but we say the DR locus, there are multiple DR genes, and I'm trying to remember now. I think a lot of people have 3 DR genes, DRB, they're called DRB1. So here would be a name. Here would be a name for a allele defined by sequencing, because we know exactly what the sequence is with our technology now. And we can do that by PCR. So something like this, it might say, so this is the locus, DRB1, and 0101. So that's an, that's a what's a thing that has many, I guess it's an asterisk.
Yeah, I see that.
Yeah, yes. So that would be how people refer to these alleles now. But originally, if you look at older literature, people speak of, say, the DR4 haplotype, and you know, that sufficed for a lot of things. Okay, so I just wanted people to be aware of that. Yeah.
So DRB1, is it still antibody or is it?
That's a locus in a gene.
So now is it only DR, or we have now DQ?
Well, no, we know there's DQ and DP as well.
And ABC.
Yes.
AB.
Mm-hmm. Yep. Yeah. So now people are able to actually look at the actual sequence, and sometimes they find fine differences that maybe aren't distinguishable by antibodies or weren't distinguishable by antibodies originally. But I wanted to give you just a background on that, how those were originally figured out. The old literature will say, well, the DR4 haplotype is associated with type 1 diabetes and rheumatoid arthritis and such. And then subsequently, so a little minutia here is why this is important, and this is some personal stuff. The guy that, the institute where I worked for many years here in Seattle, this guy had a great pedigree before he started that. Like he worked with Baruch Ben-Asaraf at Harvard, was a Nobel Prize winner, and then he became here, and he was a PI at the Hutch, and then they started his own institute. But one of the things that he was famous for is that he had distinguished the DQ locus from the DR locus as far as the importance of the type 1 diabetes association. And there was a stronger association between the DQ locus, certain alleles of the DQ locus, than of any DR locus. So that's kind of minutia, but it's the kind of thing people care about, what is responsible. So a little bit about that. But of course, know now, as you're well aware from your own work, one can now sequence, if you're talking about something where the genes, the loci are well known, it's easy to PCR and sequence a lot of people quickly, right? So this can be done to match people. Okay, but originally antibody defined. Yes?
D1 is the locus.
Pardon?
D1 is the locus.
DRB1.
And DRB1 is
Yeah, so it's DR, the DR region, and then B, the beta, and then 1 is the first of several genes. And
0101 is the
and different alleles would be given a name, 0101 or 0102. More than you need, but I think the historical relevance of the antibody defined, serologically defined versus DNA sequence defined. And this is a field, because there is such great diversity, there's a lot of minutia and lore about it. There are people that really know all the fine points. I'm not really that much into that, but people I know did that. I was telling people earlier that one of the women that I worked with, when she was, she was older than me, but when she was young, I think she was a technician on the matching team from Fred Hutt doing the first bone marrow transplant. So she knows all this minutia, and we'd always go to her for that kind of stuff. And there's interesting stuff like, one of the local populations of Native Americans that lives up in Alaska, like they're all, I say all, I heard they're all the same HLA, that's kind of an interesting thing like that. Anyway, they're all DR4 here. Okay, and again, different DRs, different haplotypes are associated with different autoimmune diseases. So those associations are known for MS, for type 1 diabetes, for rheumatoid arthritis, for juvenile rheumatoid arthritis, for celiac disease, I think for every autoimmune disease. Whenever I say always or never, you should slap me because it'll turn out to be incorrect, but that's my kind of what I think. Okay, so a little bit about that, about how we find it. So I asked you to talk a little bit about to think a little bit about what thymocytes have to do. The old, I think you don't hear this quite so much, but people used to say thymocytes are educated in the thymus. They learn what they have to do. So what are some of the things, before we actually talk about what we know, what are some of the things that the thymocyte has to go through, develop the ability to do in the thymus?
Are we talking about after they migrate to the thymus?
Yes.
Okay.
Let's say.
They need to differentiate into CD4, CD8.
Okay, that's certainly true. Yeah.
And they need to bind to self-peptides, right?
They have to. If they do, then what happens to them?
If they do bind, then for like too strong of an affinity, they die.
Mm-hmm. Okay, very true. Anything else? Anybody wants to?
To distinguish self from non-self.
Mm-hmm. So that's kind of part of that. Mm-hmm. Okay. There's also other lineages, the gamma delta lineage as well as the alpha beta. So some decisions about what kind of cell to be, whether or not you need to be deleted or not, and recognize this whole big picture. The answer to the big picture questions are really profound and amazing that our immune system does this. So create this tremendous diversity, and we touched on that already, how this happens, and then develop, and one of the methods, not the only method, but one of the methods that is used to eliminate or minimize autoreactivity is the idea that after creating this great repertoire, that some of these guys will be deleted, those that recognize self-antigens. So the diversity is created, and then at an early stage, before these are functional, it has to be before they're fully functional, right? Before they're fully functional, they can be deleted, okay? And as those of you who've read the chapter or started so far know, people have studied this in huge detail, and you don't need to know all the detail that's listed in the book. I think I gave you some help on that. Okay. So some advisories on that. So those are our main things to develop. Pick a lineage, alpha, beta, or gamma, delta, or even NK cells, and also be deleted, or pick lineage again, CD4 or CD8, and then develop your full powers of being what a lymphocyte will do. So that's a big task for the thymus, considering that people thought the thymus was just some vestigial organ that wasn't good for anything for a long time. That was cited in your book. Pretty remarkable. So one wonders what thing that we take for granted now or that we don't appreciate will turn out to be important later. But so people study the thymus for the education that occurs there. And mice especially are very manipulatable, able to be manipulated in this way, that you can transfer thymus from one mouse to another. You can look at cells within the thymus, thymocytes, and see how far they are along in this pathway. And remember, what are the markers that we follow in development of thymocytes? So just as we've talked about before, two things: rearrangement of genes, like that, rearrangement of genes. And we talked about that already as far as the order when we talked about how diversity is created. Because we have this order stuff, remember the DJ of the beta chain of the TCR, and then the V to DJ, and then we have the alpha chain and all these things. And the purpose of this, all these steps, isn't to confuse the immunology student. Well, maybe that's one purpose, but the purpose is to have checkpoints. See, everything going okay? Yeah, move on to the next step. That kind of thing. Okay. Rearrangement of genes we can monitor, and also we can monitor surface markers. That's really, if there's one thing in immunology that you've probably been aware of, following surface markers. We define these populations. In fact, sometimes it's even a little dangerous. Sometimes we look at different surface markers, and people want to find there's a difference between a cell that's A positive, B negative, C positive, D negative, and one that has one difference, and then people really can't find a difference, but they look really hard hoping to find that difference. Okay, so we look at rearrangement of genes, and surface markers. And that's kind of our scorecard, if you will, as we go through thymocyte development in thymus. And again, remembering what we have to do is pick a lineage, alpha, beta versus gamma, delta, pick a CD4 versus CD8, being a helper T cell or a killer T cell, and eliminate autoreactive cells. That's a pretty big ask for an organ that people didn't think was important not that long ago. Okay, I'm going to share my screen. And so here's our what's going on this week. Note that I did have list exclusions here, and the exclusions are not great because I wanted to tell you everything. No. So under that heading, I said, don't be concerned about the fine points of the different stages of different double negative stages, but do know overall what happens in all the double negative stages together. Also, don't worry too much about the cortex versus medulla and the thymus and different parts. I admit that's not my strong point. Be assured there are people whose careers are based on sectioning the thymus and looking at what's going on in different parts. But more focus on what all the processes that happen overall and what goes in and what comes out. Okay, I think after I added this, I added another PowerPoint link after this, and there's this one, that's just 2 slides that we'll get to in a minute. So cells entering the thymus are not even yet committed to being a T cell. They come out mature and tolerant. So it's an overview in see here our staining. As I say, there are people. There are people that is their thing to stain, to take a section like that and to look at what's going on there. And I'll have a comment at the end. I don't want to digress at the moment. Okay. But again, a diagram on the right showing these many stages. So we go from an early double negative. I want to make a comment about the statement double negative. When people are talking about thymocytes and they say double negative or single positive or double positive, they're talking about CD4 and CD8. Okay, so the double negative thymocytes have no CD4 and have no CD8. Single positive might have one or the other, double positive have both. It's a little bit confusing because sometimes if you're in another niche of immunology where maybe you're not talking about thymocytes, people might be looking at 2 markers and they might use that kind of terminology. So you should ask them, check yourself or ask them, what do you really mean there? Yeah.
What is the frequency of double positive to single positive cells?
So there's a lot of double positives and not that many make it out as single positives. So anyway, I hope we're clear, and you can challenge me as well. I think we're talking a lot about thymocytes and referring to those CD4 and CD8 markers, but I can imagine situations where even immunologists would be talking about maybe T cells in the periphery and might be looking at 2 markers, and they might kind of let that terminology slip in. It's easy to say that. Similarly, when we speak about positive selection and negative selection, we're talking about T cells in the thymus. That means a specific thing, but sometimes even in vitro, we do a selection with antibodies to remove particular types of cells or to select for particular types of cells, and it's easy to refer to those as positive selection or negative selection. But you're entitled to ask me or any presenter or any textbook, okay, what do they really mean when they're saying positive selection or negative selection? But when talking about thymocytes, this means double positive, single positive refers to CD4 and CD8 on the surface. Positive selection refers to thymocytes being allowed to be selected to move on, or negative selection being allowed to die. Okay, so we see here is overview. It has a lot of stuff on it, entering as double negative cells. As it says, these are still multipotent and they can even give rise to myeloid and NK cells. Commitment to the T cell lineage, gene rearrangement, they can become gamma delta cells or alpha betas and single positive CD4 or CD8. So all the lives, the possible lives of a thymocyte. So precursors are in the bone marrow, migrate to the thymus, and here discuss the cortex and the medulla. Begin their travel in the cortex and go to the medulla. The thymus makes fewer and fewer T cells over time. That's why people thought that maybe it wasn't an important organ, because it seems to be less active over time. So the precursors in the bone marrow go to the thymus. They're not committed yet. They're only committed in the DN2 stage. The Notch receptor, which you may have heard of from other studies, signals commitment to the T lineage. And people have worked with culture systems to understand T cell early development using things without using the thymus. Again, this is a standard way to understand something in vivo, by being able to recapitulate it as best you can in vitro. And people have tried to do this. And the transcription factor, GATA3, G-A-T-A-3, is important for relaying the signal that's received by Notch. Variety of different stages, referring to double negatives and double positives here. Positive, and they have to go through a positive and negative selection. This is our slide we've seen earlier. This shows studying lineage commitment in a dish where the Notch apparently signaling through the Notch receptor commits to the T lineage. So this can be done in vitro in one of these studies where we attempt to figure things out in vitro. About a Notch has to be present in that, I think it's referred to in the book, that constitutive expression of Notch can make everything T cells and not B cells, and knockouts of Notch can be all B cells and no T cells. So thymocytes progress through double negative stages. They arrive at the corticomedullary boundary. The Notch ligand is on thymic epithelial cells. Thymocytes get a signal. First in the outer cortex, where they get the T cell receptor and proliferate, then the medulla, then they exit at the corticomedullary junction. Again, use of surface markers to follow what's going on. So 4 double negative stages. I said don't worry about those, just know what ends up there. So some of these markers are c-KIT, you may have heard of, and people studying stem cells. CD44, an adhesion molecule. CD25 is the alpha chain of the IL-2 receptor. Touched on that in other other parts of the course. So TCR rearrangement begins at the DN2 stage. Again, minutia you don't have to know, but phenotype and how these are defined. Again, what's the status of rearrangement, and what are the surface markers? Okay. So DN1 isn't CD25 negative. Then DN2 is where it can be rearrangement of the gamma, delta, and beta. It's a T lineage commitment. CD25 positive. CD44 is lost, and CD25 is lost. So that's way too much minutia to know all that stuff, but know that people do study things like that. Okay. So in DN2, gamma, delta, and beta rearrange. Now they're fully committed to being T cells. c-KIT and CD44 expression decreases. DN3 cells that rearrange beta become alpha beta TCR T cells. These lose CD25, stop proliferating, and go to DN4. And here's another slide showing DN1, 2, 3, and 4. Eventually, so we see your pre-TCR in the surface, then a TCR in the surface, and possible positive or negative selection. Here, recombination begins in the double negative stage, so getting an alpha beta or gamma delta. It's thought that the choice depends on who can rearrange first. Gamma delta cells are going to kind of be ignored in this class. They're especially important on mucosal surfaces. The gamma deltas leave the thymus as double negatives. Many of these recognize unconventional ligands like lipids. The receptors are not nearly as diverse, and we're not going to talk as much about those, and I have much less knowledge of those gamma delta cells. And saying a lot of that same stuff. TCR beta rearrangement is much more likely to happen, and therefore alpha beta outcomes are more likely. I'm sorry for leaving that light on. I probably should get rid of that. So gamma delta are more common early on also. And suggested maybe different cues there, okay? And the book and I will focus more on the alpha betas. Book argues you're 3 times more likely to have alpha betas and because of one event rather than 2. But anyway, gamma deltas leave early. There's a higher percentage at birth, but later only 0.5% of cells are gamma delta. I've heard people suggest that more than that, and it's kind of hard to evaluate, especially if all these are associated with mucosa. Okay. So now, next in our box here, we're going to go through beta selection, which results in proliferation and differentiation. So remember the story with development of antibodies? At first, there was a heavy chain and the surrogate light chain. So it's analogous here. A lot of things are the same. So first there's rearrangement of beta, and then a pre-TCR alpha to pair with that. So what does this allow the cell to do? What's the purpose of this? To put something on the surface that then can possibly receive a signal and to say that everything's okay, we can go ahead. Okay. So I have written in my notes here, it's assumed there's a signal here that's given. It's unclear what that signal is or if one is needed, but it's thought that's what happens. So signaling at DN3 means maturation to DN4, proliferation, allelic exclusion. Remember what allelic exclusion is? We learned that antibodies, each cell only makes one type of antibody. Each T cell only makes one type of T cell receptor, although that exclusion is not as perfect in T cells. One of the benefits of this, of allelic exclusion, is for the clonal selection hypothesis, right? Where the rare T cell, the needle in the haystack that's stimulated, divides many times, and it's not as efficient if you have one T cell or one B cell making a whole lot of T cell receptors or antibodies. Okay. So now we have allelic exclusion, development to the double positive stage, and starting alpha chain rearrangement. So now double positive cells are ready for selection. So double positives are about 80% of thymocytes, answering your question earlier. Okay. So now we're up to this point in the slide with the thymocytes being subject to selection. And of course, negative selection, we need negative selection for tolerance, to be tolerant of self-antigens. Okay, a slide with lots of stuff. Let's make it bigger. So here we have, well, it's kind of jumping to the conclusion. They give away the answer here. So here we have, so current models are based largely on affinity and being able to see a ligand in the thymus, and the idea that high affinity self-antigen, as you might expect, leads to deletion. No interaction leads to death by neglect. That's a great phrase. Let's use an analogy. Death by neglect. And low or intermediate affinity with the peptide MHC leads to positive selection. Allowed to go on, yeah.
How does death by neglect work? Like, the cell just dies because it was not given something?
The death by neglect? Yeah. Mm-hmm.
Oh.
Okay, so it has to receive that signal. That's the, although it's not, it's not very well worked out at all. Okay, and we also have this. Here's a discussion of negative selection here. Let me go on with the text. So we'll discuss this stuff in some detail now. And there's some real challenges to this whole thing. So this kind of makes sense, but then you think about, what if, does this mean that all antigens have to be expressed in the thymus? What if there's an antigen only expressed in your big toe? Does that mean you're gonna have T cells that are gonna be reactive with your big toe and they get in the periphery? And the answer is sort of, kind of have a partial answer to that. Some theory completely explains everything, and it's this molecule, AIRE, given here. So, overview. Most thymocytes are double positive. Not seeing that indicates something weird. Positive selection, low affinity, and not only allowed to go on, but also gives MHC restriction, which we talked about. Only recognizing right MHC, negative selection, those having high affinity receptors are removed. So most fail. So this whole operation, this whole process is a low yield thing. And I guess maybe it has to be with our creation of such great diversity at the level of rearrangement. So again, positive selection for those that can bind self-MHC, negative selection for those with high affinity. Only 2 to 5% of thymocytes make it through. Great majority die by neglect. now it's time for an important experiment that MHC restriction is learned in the thymus. So if the thymocytes are educated, well, this is their schoolroom, or this shows what the thymus is. Yeah. I'll stop right there. So we know this is an experiment somewhat similar to what we talked about before. Another mouse experiment, another experiment with LCMV, and this is Zinkernagel again. Okay, it's interesting. I don't know what strains were used, but they just use A and B here rather than the real names of these strains. So this is an A cross B mouse. In other words, having two haplotypes. So it's a product of a cross between an A and a B. If this mouse is infected with LCMV, you're going to generate killer T cells, included in the spleen cells as shown here. And then if we're asked if those can kill targets, remember the targeting assay where you mix potential target cells loaded with radioactive chromium and then mix them with a source of CTLs, killer T cells. We look for killing by release of chromium. Okay, spin down the cells, the chromium is in the supernatant. So when this is done, these cells these killer T cells can kill both strain B and strain A. And that's expected because we expect to have positive selection on both the A haplotype and the B haplotype. Okay? Now, here's the key experiment though. Taking this mouse, this A cross B mouse, H2A, A cross B, and then the mouse was subjected to thymectomy, lethal radiation, and then it received a thymus graft. Okay, mice can be manipulated, can add back a thymus. The thymus that was added back is H2 of B, so only one of the two haplotypes, right? So this mouse is A cross B. This is the thymus is B, and also the hematopoietic stem cells that are A cross B. Okay, so the thymus is only B, the stem cells are AB, and then infect with LCMV, and now, I don't know if you can all read that. Can you guys read that in the bottom? Now, take spleen cells, having killer T cells among them, challenge and do another killing assay, and now what they found is these LCMV infected these spleen cells potentially having CTLs are mixed in with target cells of strain B. There's killing, as you might expect, but there is no killing with strain A. So our interpretation of that is that the potentially strain A restricted thymocytes did not undergo positive selection. So they're not there. So only the B targets can be killed. Okay, people follow that? So there's the potential, the stem cells were A/B, but the thymus was only B. So the restriction occurred, and the restriction was limited to the B. So clear experiment, again with LCMV and mice of two different haplotypes. So the conclusion is that the thymocytes learn their MHC restriction in the thymus. here, I guess, the results, that you should read it rather than just listening to me. Challenge with strain A, finally infected target cells, the cells were not destroyed, demonstrating an inability to select T cells recognizing strain A cells. In challenge with B, they were destroyed, demonstrating that the new thymus and bone marrow selected T cells could recognize strain B cells. Okay, any questions on that? So demonstrating that, again, that there is restriction, we knew before, but it occurs in the thymus by mismatching the thymus and the stem cells. So that's, Zinkernagel was involved in that as well. Okay. All right. So other things, as an aside, so note the ability to do an adaptive transfer here is one of the reasons that so much work has been done in the mouse, because you can do things like this, mixing haplotypes. I'd call it kind of pseudogenetics, where you have part of the mouse with one haplotype or two haplotypes and part of the mouse with only another one. Okay. So we see here the benefits of adoptive transfer and also congenic mice. Okay. So are we good with that? So now another experiment on selection and negative selection, a classic experiment, and this is done by Harold von Boehmer. I have to say Boehmer. This is one that's in the box in your text for those of you who have the books with you. I was saying to people who were here before class, inexplicably there's a few changes between different editions as far as PowerPoint's shown, and this is one of the most noted experiments in immunology, and they didn't include it, the actual data in the PowerPoint. So I took care of that. And there's a couple things I want to mention here. So again, this is Harold von Boehmer, and this might have also been done in Switzerland, I'm not sure. Note here, this is the first example, I think, of where we talked about using transgenic mice. And how is this valuable? So one of the problems with immunology is you have this really great diverse repertoire, but that can make it hard to follow any one particular T cell or B cell, right? Because you have the vast array of B cells, each of the naive animal, each making a different B cell receptor. And then even after stimulation, that population is going to expand, but still, it's still going to be a minority of all the B cells, and you perturb the system by expanding it. Okay, same thing for T cells. So there are many examples of us learning a lot from transgenic technology, but this is one of the big ones because of the fact that we can induce a transgenic T cell receptor, and because of the fact of allelic exclusion. Remember, allelic exclusion means if the transgenic T cell receptor is there, it's not going to rearrange another one. So essentially all the cells are going to have a certain T cell receptor. And in the analogous kinds of experiments with a transgenic already rearranged, so like when I say transgenic, it's already rearranged in the germline, in a transgenic B cell receptor, they're all going to have that one specificity, and because of allelic exclusion, others won't have it. So appreciate the importance of that. So that allows us to follow a certain specificity easily if all the T cells are of a certain type. Now, I hope you're thinking and you realize that's not very natural to have all the cells be of a certain type, and there's even been adjustments made to kind of allow for that. But recognize how, especially for the early experiments, this allowed a lot of things to get taken care of, to be tractable. So this is a beautiful experiment by von Boehmer, and they worked with a transgenic mouse making a TCR specific for the HY antigen. So the HY antigen is made only in males, okay? It's not made in females. So here you have a beautiful experiment in which littermates will be different in that the males will express the HY antigen and therefore HY antigen will be recognized as a self antigen, and for the littermate females, that antigen won't be expressed and therefore won't cause deletion. Does everybody follow that? I think I probably said it in an awkward way, but so that's a beautiful experiment in that way. So here's the background from the original cells, the T cells, cloning the alpha and beta cells to make the transgenic mice. And so this TCR, it recognizes the HY antigen presented by D of B. Okay. So you see here are some different mice. And so a male D of B is going to make the HY antigen. A female D of B is not going to, and neither is another female that's D of D. I don't know if you can read this, that's a different haplotype. Okay, so we'll look at the thymocytes that are found, and let me see what's shown on the other slide here. Okay, so here is a normal thymus. You see the great majority. Oh, and this is a characteristic type plot. CD4 on one axis, CD8 on the other. Sometimes you see CD4 on the X and CD8 on the Y, but this is CD4 on the Y and CD8 on the X. And these lines here are arbitrarily drawn, but it's pretty clear here. So this type of, you might have seen this kind of pattern on like a weather map. It's like about areas of pressure. So this area here, this indicates really a lot, high density, okay, when you see many lines together. And you see total number of thymocytes there. So we see here in the a normal thymus, a wild type thymus, a lot of double positives. Yeah, a little more than twice as many, but you're more CD4s than CD8s, okay? So we see that. So what happens in a male? So males now are expressing the antigen, so all the T cell, all the thymocytes are going to recognize that antigen. And I should say that the T cell receptor is under the control of a T cell specific promoter. We see here that there are essentially no single positive cells seen in, all the cells are double negative, and the double positives are, there's very low number of cells. And we think this is because there's been deletion of the double positives, because they recognize a self-antigen. So let's look here at the female expressing D, a different haplotype. So here we see double negatives and double positives, but no one is becoming single positive. So they are not undergoing positive selection and not undergoing the negative selection seen on the male D of B. Now, how about the female D of B? Kind of the key thing here is that, so D of B is the right MHC, but it doesn't have the antigen. So it's not going to be negatively selected because it's a perfect match, but we might expect positive selection because it has some affinity, because it has the right MHC. Okay? So in practice, what we do see is a fair number of single positive thymocytes in the female D of B. So, and there's CD8, and this is, I should have said, this is a TCR that recognizes CD8. Sorry, the CD8 TCR that recognizes class 1. Okay, so this suggests that the TCR MHC peptide interaction affects lineage here, leading to a CD8, but not CD4s. Okay, so idea here is that in this beautiful experiment that the self-antigen deletes the perfect match, a high affinity, the intermediate affinity with the right MHC but a wrong peptide allows some single positive selection, and the female with not the right peptide and not the right MHC leads to will eventually lead to death by neglect. Okay, so a classic experiment by von Boehmer. Questions about that? That's the second link on your home page to von Boehmer. This is goofy. You have to remember how this table, the CD4 is positive. Anyway, so you see that result. So beautiful experiment, and this is consistent with what I think it says in the book, but you might call it a Goldilocks model, that not too strong, not too little, but just right. And that allows for positive selection. So the cortical thymic epithelial cells are believed to uniquely provide signals for positive selection, while dendritic cells, B cells, and the medullary epithelial cells could all provide signals for negative selection. Again, I call that the Goldilocks model with just the right intermediate affinity. So we see all these things come together with the presentation of antigen by MHC, the dual recognition of MHC plus antigen, and the intermediate affinity allowing for positive selection. A lot of work has been done to try to figure this out in vitro systems. Still not completely sure what peptides carry out, are responsible for positive selection in vivo. But no, this is a complex environment with many interactions between thymocytes and stromal cells. Okay, I think I'll leave it at that point here. So today we talked about the overview. The overview is that all these things have to be determined as the thymocytes get educated, with the ultimate result of thymocytes being deleted if they're going to cause problems, and those that are going to be functional now being ready to go. We talked about different stages and how they can be measured, and we already had mentioned the phenomenon of MHC restriction, in that the MHC have to match, and we saw that restriction is learned in the thymus through positive selection by the experiment in which a thymectomized mice was added back a thymus with one haplotype, while the stem cells that produced the T cells were heterozygous of two haplotypes, and we saw a positive selection of only one of the two haplotypes. Then we further studied the von Boehmer experiment which is making use of the transgenic TCR. Again, an important thing that will be done throughout. Experiments talk about. It allows us to look at a situation where all the T cells are of a certain specificity. And that's very important to be able to do that. In that, we saw that the thymocytes that were specific for a particular MHC and peptide, those, when that peptide was present with the right MHC, we saw negative selection or deletion. In the female mice that had the right MHC, but not the right peptide. By the way, I don't know if I stress this enough. The MHC molecules only come to the surface with a peptide associated. They're never open and not bound to peptide. So when female mice, which now have the right MHC, but not the right peptide, there's a certain amount of positive selection seen. And we believe this is consistent with the model, the Goldilocks model of intermediate affinity allowing for positive selection. Those mice with a different MHC, say a female mouse with a different MHC that has neither the right peptide nor the right MHC, we see a lot of the double positives, but no positive selection, and those ultimately will undergo death by neglect. Okay, it's kind of a lot, but that's a real classic experiment there. Something that you might be thinking, it's also, while it's very useful experimentally to work with a system with transgenic TCRs, and people have done a lot with that, it's not very natural. And in fact, for some experiments where people want to have a more natural situation, they use a transgenic mouse, transgenic for TCR, and then they adoptively transfer a minority of those cells to another mouse so that you have enough cells to follow, but not all the cells, because it's not very natural to have every T cell in a mouse be activated. Subsequently to this famous von Boehmer experiment, some people have questioned how it was set up. No one questions the results, but they used a promoter that's a T cell specific promoter, and it's believed, I think some people said that that promoter might come on a little earlier than the natural T cell receptor promoter, and therefore that expression might be a little earlier, so that this experiment might be a little bit contrived in that way. But no one questions the overall result of the experiment. Again, a beautiful experiment by using male and female litter mates that can be male and female. They either express the antigen, the HY, or do not. Okay, any questions about interpretation or how it's set up? Okay, all good on MHC restriction? And the value, again, these really show the value of the mouse as an experimental model because of all these manipulations done. You know, it really is remarkable, all the manipulations people do. Okay, so I gave you a couple of real classic experiments there, demonstrating restriction for positive selection and also for negative selection. We'll continue next time with a bit of the in vitro work on positive selection, how that actually happens, and some work being done by a former person I knew here from UW, which is especially interesting. Okay, okay, so I think I'll stop at that point.